Come forward. Yeah, sit. We are not going to bite you, okay? Sit. Come forward. Yeah, this is. हाँ नहीं मैं. So we are on job now, okay? Yeah, so just before we start, I want to give you a recap. It's been a fascinating 3 days. We are on the fourth day probably, and hopefully you would have got something to know about drug discovery and development. And I just want to recap from the very first day you understood that what the biology is, what is the different stages of drug discovery. We talk of it. We talk of target identification. Yesterday it was touched more of a cancer, the biology, the disease, cancer as a biology disease. We all have learned something more about it. Then we understood the two arms of drug discovery. One was the efficacy or activity, which we say. Another arm, how to make that compound which should be an active, and what is the role of a chemist who is a taskmaster in that whole process, and who takes care of all, who is the center stage of it, how he tweak the compound, how we make the baby, and baby becomes the boy, and it graduate, and it's all become one of the thing which we all know well. It's saving a life of millions and billions. Along with that, in industry, sometimes it has been quoted, one of the people who are the beneficiary of a system, they take a lot of money out of it and they earn money, but there is a lot of investment. We have seen there is a 2.8 billion as sum with total of a money which is spent. But the question is, why the hell you will spend so much money if you don't get something? I have already told the Ketrada story that it makes money every year what he spent for the earn for the whole month, spent for the whole drug development. So there is a balance between it. Then we talk of a lot of the academia, role of academia, how the collaboration with industry and academia will be helpful to you guys, and that's important. Academia is the most important arm. Most of the drugs which has been there, I would have shown you a list, or sir has already told you a list, that how much the role of academia has been in discovery. You guys do the basic biology, basic discovery from the academia, and that's what help us to take the leaf out of it, and we as the industry try to tap on it, and the chemists work on it, and we all do it. So important is, there is a lot of things, there is a huge flow of information has been. I probably feel that you guys have taken some out of it, and I don't want that you all take all, but choose the thing which interests you, and hopefully tomorrow you guys can be a better researcher. You might have an aim to do anything, and that is the only aim which we have here, okay? So take one leaf out of it, that I want to do this research, I want to go to the industry, I want to do my future, and I don't want to be a very hi-fi scientist. No, we need a small stuff, and I think that will make a huge difference, okay? So now the question started, how we build up. The thing is, what is the potential pitfalls and alternative strategy in a drug discovery? The task is given to me to complete what exactly is. So I will try to cover some of the stuff before we go. So this is what the central dogma for a drug discovery is. You all know well central dogma. You know the DNA, you know the transcription, translation, we talk of a genetic term, but for a drug discovery this is how it is. You have a target selection, you lead discovery, medicinal chemistry support, and then in vitro studies, in vivo studies, and then you have a lot of, these are the preclinical, and then the clinical trials and all that goes forward. This is what you already run. Hopefully you all have remembered this slide many times. This is how we feel, you know. You all talk of telling that industry being a selfish beast altogether, people take off it. No, that's not the case. We spend money, we do a lot of stuff, and we all are also human beings. So there is an emotional touch to all of us. We all do science, and once the experiment fail, I can tell you we all get crazy, and not one person. I don't remember in my early days when I joined 2004 industry, and till 2008 I used to come home by 6. We came to it at 11, 12 sometime. System stop, we have to develop these timelines and all. It's a very tough place, you know. Industry says you work for me, I will give everything to you. I will take care of food, coffees, stay, putting up here, going there. You don't have to worry, and you don't have to run for any chemical, you don't have to arrange anything. There is a call there at a talk, you have to talk to supply chain, hey, give me this molecule. That's their headache, nothing you. You don't have to run, you have to work and focus on your work, that's all. And probably that's the biggest difference which academia and industry have, the resource and how you do get it. So this is what I want to bring, I wanted to make an image of it, just to say you 2.6 billion, there is a time ticking to it, discovery and expensive and time consuming, and it took 10 to 15 years, we all talking on these things, and then all of the sudden you feel this is an expensive, exhaustive and essential mean, you know well, but what happened where hope meets a hard reality, and that's the price of discovery. It's not the money, it's this scientist gets emotional how he tears his hair just once he gets the failure in front of it. This is a slide which tells when promise meets reality, real world fallout. So I'm giving you some example. This is Vioxx, if you see, this is a financial impact. See the financial impact. This is 4.85 billion settlement, and then 2.5 billion dollar lost sale. It happens to a company, and it was the simple reason was the cardiovascular tox. It was pulled out. Similarly, if you see Avendia, increased risk of heart attack, and this is the money which was involved. So what I want to say, you develop a drug, in the process there is a failure, fine, we all understand, and we can rectify, we can go back to the board, chemistry can go back to the board, they start making a new molecule, they will design a new molecule and provide the thing. You have a failure, fine, this is taken. But sometime failure happens when you go to the clinic and there is a withdrawal. And that withdrawal, why it happened, I am giving you some example. These are the withdrawal, failed efficacy despite acceptable safety. These are the, and there is a cost reported. Yeah, I know you guys have been seeing the newspaper of late. There is a story with the cough syrup going on here. It was exported and India causes a child death and all. That's also it was a ignorance, okay. It was a regulatory aspect which was not taken care of it. Fine. So what I want to make, these are the small pitfalls, but have a huge impact in terms of a timeline, in terms of your own image, in terms of the image to the industry, to the people who are taking the drug, and this is the financial impact, which is a great stuff for any business running places. So I will take one by one how the disconnect in the drug discovery happen. There is a drug, if you see the disconnect, there is a fragment. The biggest problem we all talking from last 4 days, I think if you can undermine, or if you can just focus on it. If I ask you, give me 2 words which you have been repeatedly listening throughout 4 days, is one is the collaboration. You are listening this thing, and that's a reason for many failure. Most of the research happened. I was in one conference and there was a guy who was Eli Lilly CEO, and he was speaking a lot on AI, he was talking of these things. But the problem is none of these pharma people put any failure outside in the market. They all do a siloed research. So similar happen in your academic. One lab doesn't know what the other lab is doing. If you are that's a small chemistry reaction, somebody knows it, and if fails, it doesn't tell I failed when I did this. If they tell everybody, people will not try it. they will do something different. So, it is important that we should be transparent, and for transparency you need a confidence. The biggest problem I feel sometimes is you do not confident of. Industry has a different thing, there is the IPR, there is the money associated. Academia, if they remove the barriers, things can change. Similarly, discovery setup, there is a siloed drug discovery, absence of feedback loops, that is what I am trying to, it is a missing communication. What happens? Try to understand the flow. Chemists make a molecule, there is a target identification, everybody decide that we will work on this target. Then you ask the biology that whether you have an assay to test or not, fine. Then we ask chemists, can we target this protein? This is how the protein structure is, can we target? Chemistry start working. Then we take a computational help to tell about the structure and how the flow of in silico biology looks like or in silico chemistry looks like. Then it goes to the ADME. I do not want to go further. There is a continuous flow of information between a chemist and a biologist. Chemists tell, take this compound, I made it, you just see the structure, they say and give them the activity back. And then there is a discussion, थोड़ा सिग्नल दिख रहा है, अगर तुम थोड़ा चेंज कर दो तो ये पोटेंट हो सकता है. अगर ये कम्युनिकेशन ना हो और सीधे डाटा भेज दिया जाए और वो केमिस्ट को उतनी बायोलॉजी की नॉलेज अगर नहीं है तो वो कहेगा या फेल हो गया, वो बिल्कुल हो सकता है तीसरे डायरेक्शन में है. अब जैसे हमको घर से यहाँ पहुँचने में हमको इंजीनियरिंग कॉलेज आना है, अगर कोई हम बावे सैयद पे भी खड़े हैं और किसी ने मुझसे कह दिया कि यार तुम एक काम करो दाहिने हाथ पे मुड़ जाना तो उधर की तरफ निकल, तो हम तो जकरिया की तरफ चले जाएंगे, हम इधर की तरफ नहीं आएंगे. हैं कोई बता दे बहुत सही पहुँच गए, बस थोड़ा सा वहीं से नेविगेट कर लो, रास्ते में तुम थोड़ा सा आगे जाओगे तो दाहिने हाथ पे चले जाओगे, इंजीनियरिंग कॉलेज पहुँच जाओगे, नहीं तो वो घूमता रहेगा. ठीक है ना. Similar is the case as the feedback loop. I am trying to tell you, there is a communication which is important, there is a flow of information and there is a constructive suggestion, and for suggestion there is a taker also which should be there. Then you have impact on the development efficiency, risk of a, so this when you do not have efficient you waste a resource, efficiency नहीं मिलती है अगर ड्रग की तो तुम्हारी रिसोर्स भी खराब होता है, डिले भी होता है और ओवरऑल सक्सेस पे डायरेक्ट वेस्ट होता है. Risk of late assessment, this is something very important. Yesterday I talked in length about the DMPK and toxicology, and then I told you about that how the DMPK and toxicology playing a role. If DMPK toxicology you delay, you make a very potent compound, you made an IPR, you have set everything, biology looks good, you have a nanomolar compound, and then you give the compound to the DMPK to test the drug ADME property, all of the sudden the compound doesn't work, so you wasted around a 6 month or 8 month, your compound is a useless, you have to go back to the board, you have to make it, and to combat the activity which you are not getting in the ADME property, you can destroy your compound and then your tweaking can change and you will not get an activity also. So, more important is early assessment of those compound as early as possible to have a feel of that compound is important, and if you get delay you are delaying the whole process or you have to go back to the board and synthesize a new compound. Need of further early collaboration, of course, we need an integration with the DMPK toxicology, risk detection and improves candidate. So, that is important. Now, I would like to tell you pitfalls in drug discovery, a small image I wanted, target identification, hit, lead, clinical, these are the stages which we have. The best example, somebody has told you that he is making, he has done a model on dementia. I am really intrigued to know about that model who was talking of, because we all in industry, CNS is something, a target, we all want to work on it, but the problem is we do not have a very good model. And see the data, high failure rate in CNS, over 90% of CNS drug fail because we do not have a मॉडल. जाहिर सी बात है इंसान की जो सोचने और समझने की क्षमता है वो जानवरों में नहीं है, वो रास्ता नेविगेट नहीं कर पाते हैं और उनका ब्रेन अलग तरह से काम करेगा, तो हम कैसे उस मॉडल को रेप्लिकेट करें, इसीलिए उसके छोटे छोटे हिस्से को रेप्लिकेट करने की कोशिश की गई है. एंड दे हैव डेवलप्ड अ मॉडल, कुछ स्विम मॉडल हैं, कुछ कैसे मॉडल्स हैं उनके पास, दे डू इट, बट नथिंग इज around 10 or 20% near to the perfection. इसीलिए बहुत चैलेंजिंग, इसीलिए कोई भी CNS की दवा नहीं है ज़्यादा, जो कुछ है वो सुलाने की है, एंड ब्रेन वर्क्स इट्स ओन वे। तो दूसरा है फेज 3 फेलियर ओवर 30%, ट्रांसलेशनल गैप, एनिमल मॉडल, दिस इज इंपॉर्टेंट। Whoever is a budding scientist, who are a biochemist, biologist, zoologist, who really want to work, I can tell you, focus of animal model. Do something new to the animal. People will take you like something. I paid to Tel Aviv University $30,000 for one model. And that was basically a PRMT CNS compound which we have given to them. That's one model, but was a niche area. If we would have developed, it would have taken 1 year to develop a method. But they have done in 1 week and sent. For me, the time is essence. So I every time say to the academia that you develop one assay. In industry, we don't want to develop assay. We want to do something, give you a compound without naming it, you give me a data, we will put it. But only thing is you should have a very strong data, you should have a very collaborative data with you. So people say then you have AI driven stratification, adaptive trial and organized. I will tell you some stories now, make you feel better, to have some interesting stories how it became. This is a story of Phyalurudine, and this is it's a very interesting story, you know. It was an antiviral drug that showed a promise in treating a hepatitis B. However, during the clinical trial, it caused a fatal liver toxicity in several patients. The underlying cause was mitochondrial damage, which had been detected in preclinical studies, and tragic outcome highlighted the withdrawal. See, this was the drug, this was the antiviral. We could not catch in toxicology study. If they would have caught in the toxicology study, we would have got something different. They were withdrawal. Okay? So important is this was something a pitfall. If we remember it, we would. So what is the take home message? Take home message is need for the human relevant model. The case emphasizes the importance of developing predictive human relevant drug testing. So what the message is for you guys? Think of it how you can do anything with a human which is more relevant you can produce in an animal or in vitro setup. That's the crux of the matter. I know it's not easy. People talk of AI and all, but AI will only be enriched if you give some data to them. So you are the main scientist. When you sit on a lab, think of those models, how you replicate that system in an animal or in a petri dish. And that take home message for the people who really want to do a good research, think of it. Get your idea, write a grant to the people, and if it is relevant to the industry, industry will fund you some amount to start with the money. Then there is another story of TGN1412, that was a cytokine storm which happened. This was tested in phase 1 trial to treat the autoimmune disease and leukemia, but triggered severe immune overreaction. That was one of the things. And what happened during the discovery or a development process, animal model failed to predict the human response. Immune response is still a challenge, although that's why when you do immune response study, we have to go, the second toxic species is every time is a monkey. Monkey is more relevant, they have more immunologically more relevant to human being. That's why we take that model. And because the study is so expensive, we don't do a very exhaustive study, and these are the things which happen. Checkpoint inhibitors are best example. You have seen Keytruda and many things you are talking of. They have been the lifeline, and now you are. So this was more important. So important was the changes in trial. The incident prompted the improvement in the clinical trial. Earlier they did it, and then the one patient died also in this trial. And that was one of the hallmark to that. There is another example of Vioxx that was it was a widely prescribed for pain and inflammation, and initially considered safer for GIT tract, better than NSAIDs. NSAIDs, you know, निमोसलाइड्स खाया होगा, नहीं खाया, नाइस टैबलेट किसी ने खाई होगी, इंडिया में तो चल रही है, बाहर बंद हो गई है, हैं? तो मत खाना कभी तुम लोग नाइस भी, हैं? खैर लोग बेचते बहुत हैं, बिकती बहुत है, सर में दर्द हो फ़ौरन नाइस दे देते हैं, हैं? तो कोशिश करो कि नाइस को ना ही लिया जाए तो अच्छा, नाइस इज़ नॉट नाइस, ओके। सो उसके बाद क्या हुआ कि दिस वाज बेसिकली इट्स COX-2 inhibitor. Sir might be knowing more, and when he went to that lab and all in those days, these were the thing which was growing like something, I know. And now people are not doing much work on it. Cardiovascular risk became apparent through the post marketing surveillance. पहले नहीं पकड़ पाए, डेवलपमेंट में नहीं पकड़ पाए, क्लिनिकल ट्रायल में भी पकड़ नहीं पाए। उसके बाद क्या हुआ, जब वो मार्केट में गई और बाहर जब गई तो पोस्ट लॉन्च के बाद मालूम हुआ कि वो जो है बहुत ज़्यादा जो है कार्डियोवास्कुलर रिस्क उसमें आया। अगर हम प्री क्लिनिकल टॉक्स में अगर हम अच्छा सीवी कर लेते, डॉग में सीवी किया जाता है, कार्डियोवास्कुलर डिजीज जितनी है वो डॉग में की जाती है और सिंगल डोज हम लोग देते हैं और उसके बाद रिपीट डोज की 5 डेज़ की स्टडी होती है, ज़्यादा देर नहीं, एक डोज देखते हैं, उसके बाद उसका ECG देखते हैं क्या है। चार्ज नहीं है क्या? ये एक और एग्जाम्पल है, BIA102474, and Sam might be knowing it because it's more from his own company earlier. तो उन्होंने फा पे काम कर रहे थे, फा एंजाइम पे बहुत Oh wow, yeah, I can tell you. No, yeah. Actually, what happened, this also comes from something known as a confidentiality thing. And there is a lot of work goes on. Like in our company, we give JBI number, JBI 278, 778. So what happened when a program starts, there is a numbering, there is a company name which goes on, and then you have a how much the compound is made total. When you submit a compound, there is a known as a compound submission form or a compound, when you start working on it, there's a lot of compound you put it. So you start making a compound when a project start, because this is every places has a different way they do project wise. Somebody knows how much the discovery project has already been moved, or in a particular project in our company, in a particular project, how much compound we have made and where we reach it. We start with a similar number. It's a sometimes chosen also. Some people start with the 700 and they do 700, 1, 702, 778. Means this 778 means they must have put 7,000 compound or 80 compound or 90th compound has made. So it has a confidentiality, but the first number usually, like this BIA is basically from Bayer number, if I remember rightly. They also write BAY also, their number, and he Eli Lilly put actually they put LI number. LI they put it, many people different name. Pfizer every time put PF number. So if you see and put a number, they will say PF, and that will be the number which they will be writing. You can understand from where the compound came. But the numbering is basically how much the compound has been synthesized. That has not much of importance. People have a different strategy so far, but this is how it is done. Anything I want to add, sir?
Thank you.
That's the only thing. There is no norm like this. It's a code, and we don't want that to spill it. Okay. If we give some name, then it will be issue sometime. Okay, 2471 neurotoxicity. This was one of the drug which was a Fa enzyme. Fa is you know well. Fa is a target which was explored way back. You know. Yeah, Fa was basically a fatty acid amide hydrolyse. पेन मैनेजमेंट के लिए यूज़ करते थे. फेज़ 1 में फ्रांस में इसकी ट्रायल जब हो रही थी तो एक पेशेंट मर गया ट्रायल में, और ये किसी भी ऑर्गेनाइजेशन के लिए एक पहाड़ उसके ऊपर फेंक देना. इसीलिए मेरी जॉब कभी कभी बहुत इम्पॉर्टेंट हो जाती है, क्योंकि मुझे जो है, मैं हूँ FIH डिसाइड करता हूँ अपने आप. FIH का मतलब फर्स्ट इन ह्यूमन डोज है, वो एनिमल डाटा से लेके हमको बताना. अब वो 10 टाइम लोअर होती है जो एक्सट्रीम टॉक्सिसिटी हो सकती है, मगर ह्यूमन एक अनप्रिडिक्टेबल चीज़ है. यू ऑल नो वेल, आई टोल्ड यू दैट विद द वार्म ऑफ ए हैंड, चेंज ऑफ ए pH, and any shake of something, if something change the structure, why don't with the building block of something will not be a stable or unstable people. That was Isaac Asimov's story. So what I want to tell you guys is unpredictable. होता क्या है, हम मॉडलिंग करते हैं, मैं मॉडलिंग भी करता हूँ, सिमुलेशन जिसको कहते हैं कि एनिमल में इतना आया है, ह्यूमन में इतना आएगा, क्योंकि इसके लीवर का रन इतना है तो मेटाबॉलिज्म इतना कम हो जाए, मगर ये होता नहीं है. आज की डेट में मैं अपना डाटा भी बता नहीं सकता हूँ, बट देर इज़ ए ह्यूज थिंग. आई एम सीइंग व्हाट आई वाज प्रिडिक्टिंग, इट इज़ आई अंडर प्रिडिक्टेड समथिंग which I was thinking that 60 or 80 we will get, we are getting activity in just 40 also. So important is the exposure and many things. So why I am saying it, तो जब फेज़ 1 में जब वो पेशेंट की डेथ हो गई, तो उन्होंने कहा कि क्या वजह हो सकती है, और ये पाया क्या गया, फा तो एक ऑलरेडी उसमें ड्रग था, तो उन्होंने कहा कि क्या हो सकता है, ये क्या है इसको, ओके न्यूरो. एंड बाद में क्या हुआ कि ये क्या हो सकता है, हुआ क्या कि लिपिड मेटाबॉलिज्म को ये अफेक्ट करता है, तो ब्रेन में जाके ब्रेन की जो लिपिड थी उसको उसने डिज़ोल्व कर दिया, और वो नहीं होने की वजह से न्यूरोटॉक्सिसिटी आई और उसका फिर विड्रॉल आया. तो हुआ क्या कि अगर आपके पास अच्छा मॉडल होता, प्री क्लिनिकल में न्यूरो का मॉडल अच्छा होता, आप उसी वक्त टॉक्सिसिटी उसकी चेक कर लेते और आपको ये इशू नहीं होता. तो आप क्या फ़ायदा होता, एक तो इतना पैसा बच जाता, दूसरा आपका वक्त बच जाता, और तीसरा वक्त के साथ लोगों की इमोशंस जुड़े होते हैं वो बच जाते, और सबसे इम्पॉर्टेंट है आप कुछ और नया बना चुके होते. अब आप इतने साल आने के बाद फिर वापस बोर्ड पे जाएंगे, फिर एक नया काम करेंगे, और देयर इज़ अ लॉट ऑफ रिसोर्स प्रॉब्लम. सो the take home message so far I have given is we don't have a good model, we don't have a preclinical model which is good enough to predict in human. This is a scope for academia, this is the biggest scope for academia. I am telling you, people are talking lot of drug discovery, scientists, they becoming a drug, they want to launch the compound, they have a patent, they say we can do it. It's still very nascent. I want you guys to sit on a lab, make a small models, that's all, and you will succeed like something. People are ready to pay you $10,000 without asking question if you have a relevant model with some proof of concept if you have. Another is a Mefedrol story. We all know well. Maybe it was a big story which was happened. It was a calcium channel blocker, and then the dangerous interaction. DDI issue was there. DDI, I explained yesterday how the DDI works. One drug work, another drug if you give along with that, it doesn't work due to CYP and all those metabolism, transporters and everything. So that was one of the reason. This was a very wonderful story where FDA was caught red-handed. Okay, and this was a story where the controversial approval happened, and this compound was cognitive improvement for the Alzheimer disease. उसी टाइम 10 लोग बैठे थे सेफ्टी की एडवाइजरी कमेटी में, और 10 लोगों ने 10-0 से इसका अगेंस्ट किया था, फिर भी इसको अप्रूव किया गया, और अप्रूव करने के बाद फिर ये इफेक्ट नहीं मिला, और उसके बाद जब नहीं मिला तो 221 में अप्रूव किया गया, और उसकी सेफ्टी और लैक ऑफ एफिकेसी की वजह से 2024 में इसको विड्रॉ भी कर लिया गया। तो यहाँ पे क्या है, रेगुलेटरी इशू है, ये पिटफॉल्स है, ये सब वजूहात हैं जिनकी वजह से हम ड्रग डिस्कवरी को हमने इतना डेकोरेटिव तो बनाया कि बहुत अच्छा है, इंटरेस्टिंग है, ये पैसा कमाने का है, ये नहीं, चैलेंजेस बहुत हैं, और जब गिरता है तो वो उसको ज़मीन पे गिरता है, फिर तबाह बर्बाद हो जाती है, बड़ी बड़ी छोटी कंपनियां तो गायब हो जाती हैं बिल्कुल सतह से। तो इसलिए ये बहुत इंपॉर्टेंट है, फूक फूक के कदम, हर डिसीजन आपका बहुत एक कंपाउंड हम जो एक और मैसेज है बडिंग साइंटिस्ट के लिए, स्टूडेंट के लिए, ये आप इतना पढ़ा नहीं पढ़ा रख लो ना रख लो, मुझे कोई फ़र्क नहीं है, आपको भी ज़रूरत नहीं है, आपको ज़िंदगी में काम आएगा, कभी आप सोचो। एक चीज़ याद रखना, बी ऑनेस्ट विद योर रिसर्च। अगर आपने एक बफर भी बनाया है और PS 7.4 अगर बनाया है तो 7.4 लिखो, 7.2 बनाया 7.2 लिखो। ये नहीं है कि हमने बनाया और हम जल्दबाजी में 7.4 लिख के चल दिया। कुछ भी, कोई भी काम आप करो, अगर ऑनेस्ट रहो, नहीं दिल चाह रहा मत करो। ये आपकी रिसर्च आज आपको गलत नहीं बताएगी, आगे सुपरवाइजर देख के हाँ बहुत अच्छा डाटा है, बहुत अच्छा पीक आ गया, बहुत वंडरफुल है, आपका कंपाउंड अच्छा, नहीं, आप ऑनेस्ट हो जाओ। वो आपकी ज़मीर को जवाब देना चाहिए कि हमने, ये बहुत फ़ायदा करेगा अपनी ज़िंदगी में। हम जानते हैं कि लोगों ने मॉलिक्यूल को देखा, उसको उठा के कह दिया स्टेबल है, दे दिया, देने के बाद उसमें तो एरर था, उसने बताया नहीं, इस कंपाउंड को उधर डाल दिया, इधर को उधर डाल दिया, जो भी किया, बाद में पीके हुआ, पीके के बाद भी डाटा समझ में नहीं आया। पीके $2000 का एक पीके होता है, अगर वो वहाँ पे एडमी में रोक लेता, पीके में नहीं जाता, हम $2000 बचाते। कहने का मकसद है कि बहुत ऑनेस्ट हो जाओ रिसर्च पे, रिसर्च से ज़्यादा ऑनेस्टी बना के लिखना कोई ज़रूरी नहीं है। छोटा काम करो, सही काम करो, गलत आया डाटा वो छापो, गलत डाटा ने बहुत रिसर्च बढ़ाई आगे। प्लीज डोंट डू दैट। आई डोंट वांट टू यूज द वर्ड प्लैगरिज्म, आई डोंट वांट टू स्पीक इंग्लिश। ये हमारा ईमान है, वी हैव टू बी ऑनेस्ट विद द रिसर्च। जो भी काम करो ऑनेस्ट, किसी को खुश मत करो। छोटा भी बफर बनाओ तो बफर ऑनेस्टली बनाओ बस, ठीक है। I think this is the basic message which you should carry with you guys. Okay, I will give you some of the quickly. This pitfall is the poor target validation. I already give you some example where you can do multi-omic CRISPR. You all know well. If somebody knows CRISPR or not, I don't know. But this is something, a technology we should know. Inadequate translational model, homogenized model, and organoid. Admit issue, early in silico and in vitro, these are the strategies people can follow, but there should be a validated method when you are talking of any of these things with respect to in silico, anything. Biomarker misuse, that's the biggest problem which is having. The two signs which I want to give you a message and name you, that you should focus. One is the biomarker and other is the model. These two things if you can do, you can go somewhere else. You don't have to think backside where you are. Data in silos, don't put your data in silo, discuss among your friends and talking to you and see यार मेरे को ये हुआ, मैंने ये जेल रन किया, इसमें ये नहीं आया, क्या हो गया, कहीं मैंने ज़्यादा कंसंट्रेशन का तो कुछ नहीं मिला दिया था, ये प्रोटीन इतनी दूर गई, ये नहीं गई, ये हम कैसे इसको निकाल सकते हैं एक्सट्रैक्ट, आपस में डिस्कस करो, कॉफ़ी पे डिस्कस करना ज़्यादा इंपॉर्टेंट है एंड यू विल सक्सीड ऑन इट. दैट इज़ अ मैसेज विच आई लर्न in many years of my doing research. Innovative approaches to the overcome, I already told you, that is just a thing which we have already done. Why integration is matter? ये क्यों इंटीग्रेशन चाहिए? इंटीग्रेशन मैं बार बार बोल रहा, देखो इसका टॉपिक भी है इंटरडिसिप्लिनरी. हम लोग जो ड्रग डिस्कवरी कर रहे हैं वो एक आदमी का काम है ही नहीं, ये सबका काम है. कोई कहे कि मैं करता हूं, हो सकता है एक लीड करता है, एक बीच में खड़ा हुआ है, वो सबको देख रहा है, वो विजनरी है होगा, मगर वो एक आदमी ये काम नहीं कर सकता. तो इंटीग्रेशन इंपॉर्टेंट है. दैट इज व्हाट आई एम सेइंग, इट विल इंप्रूव द कैंडिडेट सिलेक्शन. जाहिर सी बात. और एक और चीज है, व्हेन यू टॉक योर बॉसेस और यू टॉक ऑफ योर सुपरवाइजर, डोंट surrender yourself with your thoughts. I'm not saying you start giving backside to him, whatever, but don't surrender with your thought. Novel ideas are everything. So similarly, my boss may be one of the project leader, he may say जैन इसको कर दे और ये हो गया, ठीक ठाक है, कंपाउंड बहुत अच्छा. हम कहेंगे नहीं, कंपाउंड अच्छा नहीं है, ये हमारी नज़र में कूड़ा है, इसको टेस्ट करो, नहीं तो फेंक दो इसको. तो क्या होता है कि उससे फ़ायदा क्या है कि आपका कंपाउंड रोक रहे हो, उससे आप आगे के रिसोर्स को कहीं और इस्तेमाल कर सकते हो. ठीक है, तो देयर इज अ साइंस बिहाइंड इट. अर्ली इंटीग्रेशन एलिमिनेट्स वीक ड्रग कैंडिडेट बिफोर मेजर रिस्क, एनहांस प्रेडिक्टिव टॉक्सिकोलॉजी. Predictive toxicology, that is a failure for that, and boosted the translational relevance, that what I was talking about. Aligning the preclinical finding, that's more important, that's more of a translational science. When you do a preclinical finding, उसको अगर clinical से अगर relevancy आपको मिल जाएगी, तो ये सबसे बड़ी चीज़ है एक drug discovery के success होने का. I believe in this concept more than anything else, concept of fail fast and fail smart. See, nobody want to fail. Chemist, every time we have a fight, I belong to a department where we are said killer. The chemist has a baby, you know, वो उसको पालता है, उसको बड़ा करता है, पैदा करता है. जैसे ही बड़ा आता होता है, हम लोग कहते हैं ये बच्चा नालायक है, इसको आप हटा दो, ये बेकार का आदमी है, इसको फेंकिए, ये किसी काम आगे जाके तुम्ही को नुकसान पहुंचाएगा. मगर वो बेबी को कैसे कोई बच्चे को अपने छोड़ता है? इसीलिए हम लोग बोर्ड रूम में और कहीं भी डिस्कशन में लड़ाई बहुत होती है, समझो मारा पीटी तक नौबत आ जाती है कि ना आप इसको फेंकना चाह रहे हो, आपको दूसरा नहीं, इसको ऐसे कर लो, ऐसे कर लो. दिस आर द साइंस विच वी हैव डिस्कशन. एंड ऑफ द डे वी ऑल एग्री टू द डिसएग्रीमेंट. ओके, तो इसमें अर्ली इंटीग्रेशन आई ऑलरेडी टोल्ड, बूस्टर ट्रांसलेशन, फेल फास्ट में वही चीज़ें हैं जो हमने पहले बताया. चैलेंजेस टू द इंटीग्रेशन, ये सबसे बड़ा चैलेंज है इन एनी ऑर्गेनाइजेशन. क्या होता है कि आप डीएमपी के टॉक्सिकोलॉजी को सेवरल चैलेंजेस की ऑर्गेनाइजेशन इन साइलॉस में काम करते हो. फ्रेगमेंटेड डाटा सिस्टम होता है, हर आदमी अपना अपना डाटा उठा लेता है. अब जैसे डीएमपी का कभी डाटा एक उठाया उसने, एक्टिविटी सिर्फ देख ले, दूसरा चीज़ नहीं देखे, कोई फ़ायदा नहीं. इसीलिए जो प्रोजेक्ट लीडर बनता है ना, उसके अंडरस्टैंडिंग सबके लिए. तो हम लोग जब स्लाइड बनाते हैं तो उसमें हम एक कंपाउंड का स्ट्रक्चर डालते हैं, हम उसकी एक्टिविटी डालते हैं, एक्टिविटी के साथ उसकी नॉर्मल एक्टिविटी भी डालते हैं, एंजाइम एक्टिविटी भी डालते हैं, सेलुलर एक्टिविटी भी डालते हैं, फिजिको केमिकल प्रॉपर्टी भी डालते हैं, जैसे सॉलिबिलिटी है, पीके है, मॉलिकुलर वेट कितना है, रूल ऑफ 5 यस्टरडे, लिविंस्की रूल ऑफ 5 यस्टरडे, वो कहाँ फिट होता है. तो वो करने के बाद फिर हम लोग एडमी प्रॉपर्टी पूरी डालते हैं, फिर पीके डालते हैं, फिर एफिकेसी की भी एक स्लाइड होती है, एफिकेसी नहीं है तो पीडी मार्कर होता है, और उसके बाद एक स्लाइड में सब होता है. तो हर आदमी की नज़र में पूरा कुछ हो. प्रोटीन बाइंडिंग भी है, अब एक कंपाउंड है 90% प्रोटीन बाइंड है, एक है 99%, और सब कुछ सेम है, तो मैं 90 वाले को ले लूँगा, 99 वाले को फेंक दूंगा, डीप्रोटाइज कर लूँगा. ठीक है, फिर आगे जाके देखेंगे, अगर फेल होता है तो फिर इसको और बढ़ाएंगे. तो कहने का मकसद ये कि डाटा को हॉलिस्टिक देखो, डाटा साइलॉस में नहीं देखा जाता है. आप एक डाटा को ना देखो, सबको देखो. सॉल्यूशन दैट वर्क, ऑफ कोर्स जॉइंट प्रोजेक्ट टीम ये हम लोग की हमेशा होती है। हमारी जब हम लोग प्रोजेक्ट टीम मीटिंग करते हैं तो एक केमिस्ट होता है, एक प्रोजेक्ट लीडर होता है, कभी वो बायोलॉजिस्ट होता है, कभी वो केमिस्ट होता है, एक कोर बायोलॉजिस्ट होता है, एक डीएमपी के एडमी टॉक्स का आदमी होता है, एक आदमी जो उसमें होगा वो कॉम्पिटिशनल का होता है, अर्ली ऑन, बाद में नहीं रहता है वो। तो उसके बाद जैसे जैसे जिस स्टेज पे होते हैं वैसे तो एक पूरी टीम बैठती, and there is 1 hour, there is a huge discussion and all those things which goes in that place. Shared data dashboard, that's the way we do early risk assessment, और वहीं से रिस्क भी असेस हो जाता है कि अगर इसको ले जाएंगे तो ये इशू है। और एक सबसे इंपॉर्टेंट है कि जो हम लोग बार बार देखते रहते हैं, एक टीम होती है जिसका काम है कॉम्पिटिटिव इंटेलिजेंस देखने का। आज मैं यहाँ पहुँचा हूँ, दूसरा कंपाउंड कहाँ है, मान लो फेज़ 2 में पहुँच गया है, तब हम उसको एसेस करते हैं, ये निकल जाएगा कंपाउंड, अगर हमको पहुँचने में 10 साल अभी 8 साल लगेगा वहाँ पे, तो हम इससे कुछ पैसा बना पाएंगे कि नहीं, या कोई फ़ायदे की बात नहीं। तो हम उस वजह से प्रोजेक्ट को रोक भी देते हैं, उसको कहते हैं स्ट्रैटेजिक डिसीजन से हमने उसको होल्ड कर दिया, ठीक है। या उसका फेलियर आ गया फेज़ 1 में, तो हमने कहा यार वो तो फेल हो गया, टॉक से उसमें दिख रहा है, और अब फिर देखा जाता है क्यों फेलियर हुआ है। फेलियर टारगेट की वजह से है या फेलियर उसके कंपाउंड की वजह से। अगर टारगेट की वजह से है तो फिर तो नहीं यूज़ करते हैं, तो उसको रोकना ही पड़ता है। This is the next gen discovery vision for the future. AI, ML, still we have to write. I am looking for some of the more data to be more authoritative in saying this thing. System biology model, this is what I am emphasizing you guys to have some good model which can help you. Personalized pharmacokinetics, that is something important. That day somebody asked me a question yesterday कि इतना कुछ होगा तो एक आदमी के लिए ड्रग क्यों डेवलप करेगा। वो इसलिए नहीं करेगा कि एक आदमी के लिए, उस ड्रग को ट्वीक करेगा। सबसे अच्छा तरीका है कि अगर उसका पीके पता चल जाए कि उस कंपाउंड का पीके इस आदमी में कैसा है, तो उससे हम सिमुलेट कर लेंगे कितने पे इसको एफिकेसी। किसी ने 1 ग्राम देने पे 3000 उसका एक्सपोजर है, किसी को 1 ग्राम देने में सिर्फ जो है 300 एक्सपोजर है, तो 300 वाले को हमको 10 ग्राम देना पड़ेगा, और उसको हम, तो इसको कहते हैं पर्सनलाइज फार्माकोकाइनेटिक्स स्टडी, ठीक है। एंड देन यू हैव अ पैराडाइम शिफ्ट इन ड्रग डेवलपमेंट, दैट इज हाउ वी हैव टू डू. Important is a global high. I was telling a story when I was talking to Eli Lilly guys and said, why don't you do one thing, put a platform, put your failures in a data. I think the discovery will be more cheaper and more faster and everybody can succeed. You can come with a common platform, but from this data, if somebody develop, they can share the revenue. He said it's not so easy to work on it, and that was one of the answer, one-liner answer he gave. I know that was just a question, but still. So key takeaway, importance of integration, DMPK and toxic reduces risk, and this we can add some other department. Summary of the tools and overflows, that what we all have talked length in our collaboration is the key. This is most important. Cross-disciplinary teamwork is essential to successfully integrate the latter stage of drug discovery, and improved outcome and the cost reduction can only happen if we integrate smartly and closely. The take home message is, let's build bridges, not silos. Is it okay? And that doesn't goes in industry, in our DMPK, or in our drug discovery and development. This also goes in your labs. So for you, you have to take for yourself, sit together, discuss. Don't keep everything close to your chest. Discuss. Things should be kept on a close to chest, but science, discuss, you will succeed. I think that's all I have. If you have any questions, please shoot. Yep.
So my first question is, like we are having some toxic drugs now which failed during the clinical studies in human trials, and they are having neurotoxicity or cytotoxicity or whatever it is. So is it possible that as we have spent billions of dollars in the production of those drugs, Is it possible now that we can reuse, or we can use a certain different application of these drugs for treating cancers? As I have came across, there was a drug, thalidomide, which leads to the fetal abnormalities. So at that time, that drug was banned, and now people are using that drug for treating cancer. Is it possible now, because we are using those drugs now that has already been
Wonderful.
prepared?
A
So
good question. So you want to take it because it's a thalidomide for you, but
I have to come here.
yeah.
This is a very, very fascinating story.
Hmm.
Thalidomide was developed for leprosy. Okay, and then after some time, this is really a very, very good point you raised. Thalidomide was developed for leprosy. Then there was they started using it, and they found that there is a fetal issue, means the children were born without hand and legs. Okay, and then it was banned. Now thalidomide come back, because cancer patient, what we say, either you kill the patient or you kill the drug, right? So they said, okay, we will not kill the patient. Okay, we'll use the drug. So that started working in especially MLL, or myeloid leukemia. Uh, Now, thalidomide, you might not have seen that this is the most important part of the protein, or protein degradation part, because this is the one which goes and binds into E ligase enzyme, okay, and we call it CRBN. That is the thalidomide, or its analog. There are two we use. There are many more. So that was so. How you said that? Okay, can the drug be used for which is very toxic, and be used for different things? This is the one which is very important, has become very important, nothing to do with toxicity, because if you use that alone, it is toxic. If you connect it something, it is not toxic. I gave an example of calcisiamycin yesterday. Very toxic, is 0.0007 nanomolar. If you give, it is toxic even at that level. But when you connect to something, it becomes better. So yes, the drugs which are neurotoxic or cytotoxic can be used for some disease, but you have to do a lot of work on that. Okay?
Yeah, that's what yesterday so I was telling it. So I asked him to tell you a story. But one more thing I can add you when you do a toxic study.
There are many drugs like
Yeah,
that. We don't have time to
and
tell you.
thalidomide, if you handle thalidomide, you can't stay in a lab for a long time. So that's having such a smell. I worked with a guy who really worked on that thalidomide. He was a Pfizer guy. He came and taught me some assay with something, and he was making. I want to tell you a story with what we do. When we do a tox study or a human trial, there is a question that whether it should be given to pregnant women, where it should be given to females, whether it should be given to the. So there you have an option to tell, no, we will not treat it. There we will. So these are also the options if you understand them. And the second most important is, more important, you gave example thalidomide, we gave a story, but in a generalized form. The story is simple. You have to understand the tox. Is it a tox coming from a target, or is it a tox coming from a molecule? Fine. If it is target for the target, fine, that target may be useless. If molecule is not there, you can check with the other target, do a quick study which is known as a bridging study, and then you can move forward. There are many drugs which have been, people are doing a repurposing. That's the reason, even with a successful and an unsuccessful drug.
So, for example, in case of calcisiamycin, it was gone after 7 years on the market, but that was a mistake by a physician. There is no physician here, right? No doctor. Otherwise, they will they will tell me something. So it was a mistake because they used to give a very large amount of drug one time, once a day, injection. Now somebody said, no, this drug is very, very effective, although it is very toxic. Okay, why not we give at 6 hour interval? and they started giving at 6 hour interval. That drug is still in market and making billions of dollars. So, you know, cancer drug, very toxic drug, cancer drug, can be modulated by little bit of, thinking into a very nice product, and it is treating a lot of patient. The survival rate using calcium is very high. That is another one. Yeah, it was used long time ago in World War One in for syphilis, and World War Two also. Now arsenic is being used for some other purpose also. Yeah, yeah.
Anything else you have? Yeah, go ahead.
Sir, during clinical trial, how you handle the volunteers, sir, in phase 1 or phase 2?
Very tough job. Very tough job.
How that how
See,
sir,
when we do a research in our clinical trial in my company, we do with the patient actually. But patient also, it's tough, you know. They are already emotionally stressed. They suffer a lot. There is a lot of people who are giving. I do with lung cancer. So people are terminal type. They are told that you will alive for 15 days, you're 10 days or something, and they come to the trial because they don't have any option outside. Then they get a lot of suggestion, falana baba hai, unke paas chale jao, falani cheez uske. It's tough. But the only thing is the doctor compliance or doctor confidence bring them to inside, and we try to save. And many of the people, one of the patient, I will tell you a story for my drug, and it's in US. There is a guy, startup is our physician. There was a guy who came. He was a 54-year male, and he came and he was said that you start preparing yourself. You have another 1 month. Okay, and then he came to us, and we startup asked him that this is one of the trial going on, and that is more of STK11 story I'm telling about. So we told that okay, you can take it and just you can move forward already. All of a sudden, that drug, that patient came to it, and today it's around 2 years, 4 months. He is still on the drug. Now he is going on vacation. He has a kid. He dropped his son day out and day out. He is normal. Only thing he tells that I pray for you guys and you keep on giving us compound. Okay, so this is how it is. We made a difference at least for one. There will be a lot to come. Yeah.
Because sir, for some disease you need 10 to 200 patients having the same disease, and we are checking for a drug. So we need patient which is having this disease with which we are working, or you are
yeah, 788, 800, 900, 1100 type of a cohort studies are done, you know. So that will take 10 years, 11 years, you know. Okay.